Everyone, I'm Chavi and welcome back to the sixth week in the Data Science webinar series organized by Manav, the Human Atlas Initiative. In some of our previous talks in this series, several of you from the audience have asked about the role of machine learning in analyzing genomics and transcriptomics data and its real-world applications. To help us understand this aspect of data science better, today joining us is Dr. Anguraj Sadanandam. Dr. Anguraj is an associate professor at the Institute of Cancer Research in London, and he leads the Systems and Precision Medicine team. Over the years, his team has done some very interesting work in using ML-based techniques for subtyping of several cancer types, development of diagnostic assays, and all the way to testing personalized treatments. In the current scenario, they've also applied these strategies to study variations in immune systems across COVID-19 patients. He has published his work in several high-impact journals, and some of this work is what he will be sharing with us today. Now, before we start, I would like to mention to our audience to post their questions only in the Q&A tab below, so we can ask them to Dr. Anguraj at the end of his talk. So welcome, Dr. Anguraj, and over to you.
Thank you, Chavi. And first of all, I'd like to thank the Manav team for inviting me. This is very exciting. It's a strange time that I cannot meet all of you, but it's very exciting to give a talk for the students and faculty in India. This is a great opportunity to virtually meet all of you. As you can see the title, When and Where to Divide to Conquer, isn't that we are all in the same situation where with the COVID, we are with the social distancing, we have been divided, and to conquer the virus, obviously we have to follow this because that's how this works. But we have been using the same strategy for cancer to divide and conquer, especially using the machine learning approaches. And I'm going to share some of those that we have successfully used to see how this would be applicable in the clinic over past 10 years. And I'm just going to show also how that's been recently been applied by us for COVID for certain questions to answer. As you all know, for whether cancer or COVID, molecular biology is the basis of it, and it's evolving fast over several decades. And obviously that's because of the technological revolution, starting from the Sanger sequencing, for example, to PCR. But until 2006, as you can see in the graph, that it was just slow moving. But now, since 2006, especially in last few years, it has been going more or less exponentially with increase in sequencing technology, mass spectrometry, single cell sequencing, multiplex sequencing, and probably have left out quite a few different technologies that are important. Obviously, what is the consequence of this revolution in this technology? It's just pouring loads and loads of information that the human mind cannot really comprehend in one go. We need the machines to obviously mine them. The omics I'm talking about is not the genomics or proteomics and metabolomics. It's now even the age-old, the pathology slides and the CT scans like radiomics information are being mined, and all those images are pouring, and it is the time we all need to integrate them to understand at the systems level what is happening. So these all omics data would not make any sense if you don't have any phenotypes, and we realized it several years back, and we always use this along with the phenotypic data that can vary. For example, the patient prognosis on the left-hand side, you can see, and the treatment responses associated with that. The drug response data from thousands and thousands of cell lines that just being put in the publicly available database, or even some of them we do. Preclinical trial data that's available, as well as the pathways. Now the whole task is to integrate. Obviously, it's not just going to be possible by simple means. For that purpose, we have interdisciplinary team within our lab, or we also collaborate widely, especially with the clinicians, to achieve some of these stats. But there are quite a few labs which are highly encouraging. So for this purpose, several years back, we put together an engine which we call Cancer Engine, which basically takes the patient samples, generates omics data, or get it from publicly available data, do the machine learning approach, the data is input for the engine, the machine learning and the processing of data integration to identify clusters or subgroups of the cancers to ask two major questions. Can we really find who live longer, which is a prognosis? And the second question is that, can we treat the patient with the right treatment? Because most of them, unfortunately, do not get the right treatment because the information is not available. We also test them using the mouse models and animal tumors, as well as the cell lines, which is a process to identify a way to treat patients by developing the appropriate tests. So often with all these information, still we are super confused where to go, which direction to go, especially from the clinician's perspective. And this is the mantra that we have: know your patient. So that means individually understanding the patient's genome or the integrated omics information probably could help us identify. That means clinical question is the first for us. Obviously, in order to understand these patients, we need to know what their cancer contains. And you know probably that cancer is highly heterogeneous, that even if you take, for example, colorectal cancer, they vary from one patient to the other, as has been represented in different colors in this schematic figure. That could probably be because of multiple factors from genetics, epigenetics, and whatnot. The conventional medicine basically diagnoses for example, colorectal cancer patients the same way and treat them the same way as well. That way, the patients are not getting the right treatment. So that has been changing recently, and we have also contributed, and there are many other contributions happening around the world. So that means the overall idea here is that one size doesn't fit all in healthcare. That means individualized or personalized medicine is very much needed. And how does that look like? So personalized medicine means match patients to therapies. If it can be diagnosis, but here we are fitting to the therapies. And in that case, certain patients do not have an aggressive disease. In that case, no treatment would be a possibility. There are patients who are highly aggressive, which is shown in the bottom right corner, which shows intensified therapy is needed, a combination of several chemotherapies needed. But there are also patients who lie in the middle with the targeted therapies and immunotherapies and whatnot, which varies from one cancer type to the other. How do we do that? Multi-Omics could help us do such process. However, the tumors are not that simple. They are highly heterogeneous in the sense not only that they vary from one patient to the other, which I showed previously as well as in the slide. They also, within the same tumor, there are differences within cancer cells, for example, which represents the intratumoral within the tumor changes. That means when you treat a patient, maybe one patient over here, probably would get cured because of this therapy. Others probably do not. For example, this one especially would make the cancer worse with that treatment. In that case, personalized medicine is very much needed, but it doesn't stop here. There is more complexity coming from the stromal and the cancer cells, which interact together, and then also the immune cells. Immune cells comes in a different flavors, from neutrophils to specifically innate adaptive immunity and many other things, and it is super complicated. Again, it is more complicated with the point that the tumors which are originating in a particular organ, it doesn't stay there. It just metastasizes, creating a spatial heterogeneity or temporal heterogeneity that's time-based. That means based on the treatment, these tumors could change their characteristics, leading to differences in the, from one time point to the other. So I have introduced all the complexity, and now it's time for us to go and study these heterogeneity together in a single patient or a group of patients. Obviously, it's easy to say than do. And obviously that needs to understand the disease, organize it more like ecosystem where there is a map already available. Again, that's not going to be, it's a long-term vision, and that requires artificial intelligence and machine learning approaches, use as much data as possible so that we could achieve this. And we all need to do this one way or the other together. For that purpose, AI plays a major role. Artificial intelligence is an umbrella term which involves machine learning and deep learning methods. And artificial intelligence means that it behaves more like a human brain. Machine learning approach means the machine relearns and learns again to provide solution that would be more useful. The deep learning, obviously, again, a subset of machine learning approach where you apply more sophisticated methods, which I will just introduce in later slides. Is AI achievable? For example, Siri is one example. Since 1956, AI concept has been introduced. Self-driving car is another example. How does it work in cancer biology? There are many projects going on. I'm just putting an example here. How about surgery and other aspects of it, which could probably be achieved through AI? Machine learning on the other side, actually, there are many projects which are going on. Obviously, there are more hype about AI and ML. Please watch for it. It's what is possible and what not is possible is very important. The whole idea of that is to see how we could achieve it such that it goes to the real life scenario. In simple way, machine learning can be divided into supervised and unsupervised. I have listed few methods. There are many other methods available. I listed those which we routinely use in our lab and are in the biological scenario. For example, support vector machine, SAM analysis, PAM analysis, supervised. The unsupervised include non-negative matrix factorization, so on and so forth. I'll also introduce some other unsupervised methods which are complicated for integrated analysis. These methods can come in, again, different flavors. Most of them are linear modeling, but obviously, I would say not mostly, less or linear models. When it comes to the integrated analysis, most of them are non-linear. That means different mixture models are needed to perform these kind of machine learning approaches. Obviously, deep learning, neural network is becoming very famous recently and applied in biology in a lot of ways, especially image analysis and other things, where you have an input layer and multiple hidden layers, which provides a solution. Again, the deep neural network can have a linear and non-linear combined together within a single model. This is an interesting paper which talks about the evolution of different deep learning, machine learning approaches. I'm not going to go into the details, but you can see since 1940s there has been a huge evolution of these methods. In the next slides, I'm just going to talk about some of the supervised and unsupervised machine learning approaches that we have applied from quality control for the data analysis, which is very, very important. If you don't have a quality data, then output is not going to be very, quality as well. And then how we applied for prognostic and predictive. Prognostic here is how long the patients live. Predictive represents which patient to treat with what. And also we have applied to different combination of omics data, which is unsupervised. So start with the supervised machine learning approach. First is a quality control, the batch correction. If you're familiar with the generation of omic data, from the time you acquire the patient sample to do the omics analysis, and then they provide the data, there are a lot more batches of samples and data being provided, which probably could confound the final results, and that needs to be corrected. For that purpose, there are until recently, the best way to do that is visualization. Probably you're all familiar with principal component analysis, and this is an example where two different data sets which are represented by different GSEs and a biological variance of that tumor and normal represent different symbols usually gets visualized like this, where you can see that these two data sets in green and orange are all highly separated from each other, representing that potentially there is a batch in this. However, this particular orange one also has this tumor and normal. Now, this is very, very tricky that we need to remove the differences in the batch between these two, not removing the difference between the tumor and normal. These kind of visualization methods, and there are correct detection methods like COMBAT, which is a standard method available, it's good, but we would not know what we have corrected and what we did not correct. That means batch diagnosis is very important. So based on that, quite a few years back, we just developed a method called exploBATCH, which contains FindBatch, which is a diagnostic tool which quantitatively assesses from each principal component, and this is based on probabilistic principal component analysis with covariates. The covariates would be the batch information. It is a supervised way of looking in to ask the question that how many of these principal components are affected by this batch, so that we can remove it. At the same time, this is a batch based, and here is the normal or tumor effect. It is very tricky when the same principal component is associated with the tumor biological effect as well as batch effect. However, our tool works in a better way. We have within the exploBATCH correct batch, which corrected this, and you can see that we brought these together without removing the tumor normal. How do we know that? As you can see here, we are showing that we are showing here that the batches are getting corrected. Batch in the normal tumor effect has been evaluated, while the blue ones are batch which got corrected. That means they're all moving towards zero, whereas in the case of the orange one, which is in biological effect, which still stays there, representing that we can correct them without affecting it. So we have this tool in our GitHub, which is freely available if you wanted to use it. So with that, once we correct the quality, then we need to go and find a way. How do we, when and where to divide to find a way for personalized cancer treatment, and how machine learning approach would be helpful there. Here I'm going to discuss the combined unsupervised and supervised methods. With respect to the unsupervised method, you find the clusters, and then with the supervised method, you basically find the biomarkers or the features that would distinguish those classes. We have published quite a few papers along those lines, starting with pancreatic cancer several years back in 2011, which has become a standard in the field. That we earlier used about 70 patient samples to divide them into 3 groups by unsupervised non-negative matrix factorization machine learning approach, where we could find 3 groups with the heat map showing blue represents increased gene expression, red represents red represents increased expression, blue represents decreased expression, and you can see that there are subset of genes which are highly expressed in each of these groups. We named them as per biology. That classical represents more epithelial-like, quasi-mesenchymal representing more mesenchymal, and exocrine-like looks like more the pancreatic normal tissue-like. We also found that by this Kaplan-Meier curve, where the proportions of patients living are shown on the y-axis and the overall survival on the x-axis, and you can see that the quasi-mesenchymal patients were living shorter compared to the others, representing they are the poor prognostic or patients who are in vulnerable situation. At the same time, this is for the prognosis, but how about treatment? At that time, we didn't have patients who were treated with the gemcitabine the data. So we used the cell lines, we used several cell lines which represent either the classical or quasi-mesenchymal. We treated with one of the standard chemotherapy in the clinic, even now, gemcitabine, and we found that this is the value which represents how these cells respond to it, which is called IC50, and you can see that the quasi-mesenchymal cell lines were responding more. Interestingly, we published this in 2011. In 2015 and 2016, papers came back and confirmed our validation. Our work validated it and showed that it is relatively true that these patients, which they renamed into different subtypes to show that they respond to the chemotherapy. Here we have 62 gene signature, which we have developed into clinical assay that is being tested to see how it would be useful in the clinic. So from that evolution of the method that we used, we wanted to apply that to other cancer, which is colorectal cancer, where we wanted to see how the same mRNA or gene expression profiling can be combined with the drug response, this time purely with the patient samples, to understand how, again, we can divide and conquer. Divide and conquer here means identifying clusters and then understanding what these clusters mean. So along those lines, we used about 450 patient samples from colorectal cancer, use the same non-negative matrix factorization method, which is one of the popular methods. And then we identified 5 groups similar to pancreatic, but we found that these represents the individual cell types that are present in the normal colon crypt. And we found that in a supervised way, which is interesting, and we named them enterocyte transit amplifying stem-like inflammatory goblet-like. And again, when we looked at this in prognosis point of view, the stem-like, which would be equivalent to the quasi-mesenchymal in the pancreatic cancer, showed the poorest prognosis. Which is very interesting that these 2 groups, which are the green one represents goblet and the pink one represents TA, these patients were living longer, even 120 months, representing that they probably would not need treatment when they are in the early stages. But this is not the case happening, and I don't have a slide here, but we have also seen that with the treatment, probably these patients could drop. So it is very important to, with the machine learning approaches, to divide these to understand. I mean, most important is that we should not be treating patients who would not benefit out of it, or if that would affect them by toxicity or side effects. However, long story short, we put together a paper which published again several years back, 2013, showing how this is one of the first papers showing boldly that how we could really classify the colorectal cancer into different groups. Again, overlaying the cetuximab, which is anti-tumor drug, we divided the transit amplifying into cetuximab sensitive and resistant, and then we put at the different stages which therapies would be useful for these patients. Actually, we are very, very happy. Even in the recent ASCO meeting for the medical oncologists, we could really see, for example, the stem-like patients are responding more to the irinotecan-based therapy. We have already shown this with the other this transit amplifying subtypes. So we are very happy that our earlier findings are becoming more valid now with the larger clinical trials, especially retrospective studies. And we developed this into an assay called nano-CRC assay, which is used now. I think we have used it for more than 2,000 patient samples globally from different clinical trials that's being reported and published. This is a success story that we just wanted to show from the point of how from the bench to the bedside through the machine learning and the other approaches, we could really see some potential benefit for the patients. Obviously, it has to go through multiple other ways before going into the clinic. Obviously, probably I have provided too much information. This is what exactly I'm saying. We have now been overloaded with a lot of information for cancer, and obviously, it's very difficult for us to comprehend. Machine learning is needed. So, hope I have convinced you that it is possible with the machine learning approaches to divide it and conquer. But as I mentioned in the previous ones, if you have noticed, there were multiple analyses that were involved one way or the other, and these analyses were mainly involving the omics data. Now, the question is, how do we bring in the clinical as well as the omics data together to provide a functional significance, that is biological meaning, and how do we avoid multiple steps, and can we do with very few steps or single step? So genome-phenome integration is very important, and we call this functional subtyping or actional subtyping. And we have developed a tool called FENMAP, which is factor analysis based method. So the idea here is, for example, if we take one omics data, let's say gene expression matrix, and then how do we combine that with the clinical information such as age, grade, stage, mutation, and any other parameters, which you can call phenotypes or covariates, to reduce the dimension and understand how we could find different clusters or subtypes, which would have all this information together within that particular cluster. So this is an all-in-one tool which can identify not only the clusters, which are subtypes, but also immediately provide the association of phenotypes, and then also signatures, which is important for the clinical purpose as well. So here is one example from breast cancer data that we modeled, where we got 36 breast cancer cell lines, but we can apply for hundreds and hundreds of samples. but here is a simple example where we use the drug response as the covariates from these cell lines. So we have quantitative information from 4 different drugs represented here. We chose about 1,000 genes, which are highly variable from these 36 breast cancer cell lines to model them. And here are the results from the FENMAP, where initially we asked the question, how many number of optimal subtypes you can find. For that, we use the Bayesian information criteria, and we found that there are optimally 5 subtypes available. And you can see these 5 subtypes similar to the principal component analysis. This is meta-variable 1 and 2, but you can have many number of meta-variables which provide different contexts. And you can see here how the density plot for those are showing. And there are these 5 subtypes. Earlier, we ourselves have published showing that by simple gene expression without this the FENMAP analysis, 5 subtypes, more or less they seem to be overlapping with these. However, we found that some of these subtypes were splitting. For example, here, these 2 were initially where we did not report them. At the same time, the same tool provides you which of the drugs would match with the clusters of subtypes we identified. So if you overlap the figure C on B, etoposide, for example, would be more relevant in the green group, which is the claudin low over here. The fascaplycin, which is a CDK4/6 inhibitor, which is also available in the clinic, which would be more for these luminal patients. And similarly, there are genes which you can overlap here, and you can find that, for example, these are the genes which would distinguish these from the others, and these are the genes which would distinguish. So this way we can really model them to identify drug, gene, and their phenotype information in one go. However, how about multi-omics? That was a single omics analysis. How about with mRNA copy number? I mean, there are quite a few tools available there. But we also developed a tool which is we call iFENMAP, which integrates. However, I would like to go over the existing tools and how our tool versus others also perform. But all these tools can be used depending on the question in place. So no tool is best. It all depends on how you apply them. So, for example, I divide these into 2 different methods. One is called discrete integration. That means you're bringing these information separately and then trying to process them separately and integrate them. Whereas a direct integrative clustering, for example, FENMAP type work would directly integrate all of them so that you don't lose the statistical information. So TCGA, if you're all familiar with that, Cancer Genome Atlas uses something called cluster of clusters, which I will explain. And then there are other methods which I will go one by one. The blue represents those which have been developed by our group. So moving on to the cluster on clusters, how cluster on clusters in simple way works is that if you have gene expression, methylation, microRNA of any type of omics data, you cluster them individually by whatever method. For example, consensus hierarchy clustering, that's what they used here, a simple method, and then take these clusters and then cluster them together to find, for example, 4 groups they identified. So though it is an interesting method, this has been published extensively in multiple Nature papers, but it would not provide you more information than what has been already available. For example, it was just providing same information that was published for the breast cancer here several years back. So it would provide you some more information, but for example, there could be 1% of patients. That means 1 out of 100 could benefit out of a particular treatment. That cannot be identified by these kind of approaches. So it needs to be evolved in multiple ways. You know, there are other methods we and others are developing. So one of the other methods that we developed for this similar discrete integration it's called iPolyCluster, which is evolution of a method that we published in 2018 called polycluster, where you can just, for example, here is a pancreatic neuroendocrine tumor that we published several years back, where we used the microRNA and gene expression. We, again, similar to the cluster on cluster, but different from that, and more using network-based integration. We showed that, you know, for example, the clusters, which are 4 clusters coming out from the gene expression, could be integrated with the microRNA clusters to show that there are 3 major clusters which are coming out. That immediately we can see a clinical importance, because when we compare it to the grades, which are the clinically prognostic way of looking at these patients, or even for treatment purpose, we can see how they are associated with it. There are other examples, some of them we have published as well. Moving on from the discrete analysis, there is a direct integrative clustering method. The popular one is iCluster, that's existing for several years now, which actually takes in these multiple omics data, integrate them simultaneously to find clusters, and potentially you can again group them into subtypes. This is actually from their publication, where you can see iCluster compared to the other methods, SVD, and where a methylation alone or expression alone can be clustered. But when you cluster them together, you can really see nice separation of the samples. However, there are more evolution of these iCluster methods are coming out, including what we have developed. So we decided to develop not only the multi-omics, but also multi-omics plus phenomics information. So here, that is what is iFenMap. So the difference between iCluster and iFenMap is that, FenMap introduces the phenotypes along with the multi-omics data that can be modeled. So this is how the schematic looks like, not only phenomics data being clustered, but also the clinical covariates of any numbers that can be brought in. So here is this very simple example, again showing that we identified 5 subtypes by bringing in methylation, gene expression, and microRNA data, and you can see here that there are this is a 3D meta-variable plot where the clusters are separating nicely, and when you overlap this, for example, grade 3 patients are all here. This is breast cancer data, so progesterone receptor patients are sitting here, so you can just relate them, and also you can find genes which are genes or methylations and microRNAs and whatnot can all be correlated to this, so those information also comes out of these. This is a very simple example, but we have used this for multiple purposes, but for this talk I'm just giving it in a very simple way. However, deep learning methods are useful as well. Obviously, deep learning are more, as I mentioned, are involved in image analysis, which we to a certain extent doing. However, our other colleagues are doing. There's also a recent Nature Medicine paper from ICR just last week showing how deep learning methods for histology images could be used to understand immune-related changes in cancer. So due to time constraint, I cannot go into the details, but I just wanted to mention that that's another area which is of high importance. Given an India context, we are working with Indian collaborators and researchers, as mentioned below here. Main 2 different cancers. One is gastrointestinal cancer, which is of our main interest, as well as triple-negative breast cancer. And there are a lot of activities going on, and we are involved in larger projects with ISO Pune, which eventually will be knowing. And this is all putting together how we could contribute to India, and we are seeking opportunities to collaborate with more researchers from India and Asian countries. For this purpose, we have put together a consortium which is being developed. It's called Cancer Engine-Based Genome-Phenome Integrating Consortium, CACPIC, which basically uses our cancer engine and collaborators to find how we could conquer this devastating disease. To summarize this part, subtypes are context specific. That means they vary from data set to method to whatnot. Especially in these cases, the patient is the central point from which we are all thinking, and that should be the central point. So, the functional and phenotypic criteria both need to be important in identifying the subtypes. Our features would help us to identify who should be treated how. And there are other tools and methods available, and confounding factors related to pathway analysis can also be identified from these. So With this, I just want to finish our recent adventure with the COVID. I have never worked anything other than cancer in my career, but obviously, like everybody else, what we could contribute to conquering Corona. Obviously, I don't know anything about virus, so we didn't work much on that direction. But the group of researchers from India, Canada, US, and other places, we all got together to find a way how we could move, do some research which probably would be useful for the community. For that purpose, again, based on the cancer engine, we put a COVID engine, which basically does a systematic analysis from patients, the blood-based or the PBMC data, which we were lucky to get some of those data, and to not only identify disease stages, which is what the divide and conquer. That means patients who are in the acute stage potentially dying, and patients who are in the recovering stage, to genes, pathways, subcellular network, immune cells, disease symptoms, as well as drug repurposing, while genes to the immune cells represents more the systemic changes that these patients are going through. And we were successful, and we could potentially do that in less time. And obviously, we published a part of this in Research Square, which is available for all of you to look at. Not going into the details, this particular analysis provided 290 gene markers, which can identify acute versus recovering patients to many other immune changes happening in these. And it's amazing what blood can provide with less intervention. This can be tested in the patients. Obviously, it's just an initial study. This needs to be rigorously validated. Very, very, very important. For that purpose, again, we have formed a consortium called CAPSER, which is a consortium to assess blood-based biosensor and artificial intelligence platform for system-wide individualized response. Again, this involves people from many countries, including India, and many biorepositories that we are trying to involve. If you're interested, please contact us. So with this, I'd like to thank, obviously, my lab members, without whom this would not be possible. We have a mix of interdisciplinary researchers from statistics, clinic, and most importantly, animal modelers and others who all put together. I thank my past and present members. Of course, collaborators are very, very important, as you can see, mainly clinical collaborators that we have who provide samples and other information. For COVID, obviously, these are all the collaborators and CAPSER consortium members who are contributing to it. Now we are more interested in studying COVID and cancer because that's because of the cancer vulnerability due to COVID scenario. This is very important. Obviously, funders, very, very important. Without them, this would not be possible. Finally, I end with the slide that we're looking for postdoctoral fellows and our bioinformatician currently from my lab. And also we're looking for MSc project students. We have my website down here, Twitter and other things. Please contact if you're interested. Thank you.
Yeah, thank you, Dr. Anguraj. We have a few questions coming up. So I will take them one by one. Yeah. So one of the questions is, do we need the whole genomic data for the patients suffering from cancer?
So if the question is referring to the point of mutations, the copy number, and the question is if it's related to machine learning approach. Yes, machine learning approach can take in as much data as possible. You will not know how much of data is needed or how much information or mutations needed to diagnose a patient. So initially it would be better to include as much information possible. That has been done, published by several groups, TCGS, consortium, many other things. But obviously by the end, probably you don't need all of them for diagnosing your patients. Hope that answers the question.
Okay. A related question is, when do you decide to use cell lines versus patient samples during the entire process of the in the pipeline?
Sure. So for us, obviously, patient samples are the starting point. We have also done cell lines as a starting point, published earlier. So it's a very good question. But because cell lines provide information how to treat patients, in the event that you cannot have patient samples, and cell lines would be a starting point. However, cell lines are useful, or even mouse models for that sake, would be useful once the patient-based data has been mined, hypothesis has been generated, and validation can be done using these models.
The next question is, it's a two-part question. The person wants to know that one of the issue of commercialization of personalized medicine is confidentiality, like who gets access to your genetic information and other things related to that. What is your opinion on that? And also, if someone knows that they are predisposed to certain disease, is it going to have any potential consequence on their mental health?
Okay, can you repeat the first question again?
Yeah,
Sorry.
so the first question is more about in terms of privacy I'll just repeat it. It says that one issue of commercialization of personalized medicine is confidentiality, like who gets access to your genetic information. What is your opinion on that?
Of course. When it comes, so there are a lot of organizations, actually a lot of information available out there. For example, from Europe, GDPR is one of those things, and obviously when it comes to the research, the patient consent is very important, and it is private. That's why researchers do not get all the information about patients and other information. So it's all de-identified. I agree that there are many other regulations, but this is a larger topic. This could be discussed in a different context. And whoever asked this question, I'm happy to discuss with them offline, and also put in touch with people who could provide more information. And what was the second question?
Yeah, the second question was, does knowing about a predisposition to a disease have potential consequences on mental health of patients?
It's a good question, but that's beyond my understanding and research. So I don't have information for that.
So another question is, how are the potential drugs identified for the studies?
I mean cancer?
Yes,
right.
Yeah.
So for the cancer, so far what we have modeled is how the standard of care or existing therapies, for example, for colon and pancreatic, can be personalized, because that's a major question. Maybe I didn't mention here, for example, chemotherapy provided for colon cancer patient, only 40% benefit out of it. Other 60% do not. Most of the therapies are like that, and there is no way to really find who will respond or not respond. From that perspective, we model specific drugs. So when I said clinical question in place, that means who are those patients, what kind of therapies they would be eligible for, and modeling more towards that could provide the way to find the personalized medicine. That's one way of looking. Another way is that you could model cell lines, thousands and thousands of them, find an indication, then go back and ask from the patient. So there are multiple strategies and approaches which could help to find the drug information.
Okay. We have a lot of students joining us, and a lot of them have questions with respect to what kind of programming languages will be useful for them if they want to, take their career in this direction of personalized medicine.
Sure. For some reason, I'm a biologist, but started with C++ and Java and many other things, but I hardly use them right now. However, I'm only going to tell what is applicable in the field right now. So R or MATLAB is the most useful, because there's a lot of statistics that's going on with these analysis. Definitely with the sequence analysis, Perl, Python, obviously Unix programming, especially learning how to use the high performance computing, those things would be useful. However, if you're developing any standalone software and other things, obviously Java or other things would be useful as well.
Okay. So there's a question asking, is subcellular changes in data also relevant, and is that captured in any form?
Sorry, can you repeat that?
I think the question is that is the changes in different organelles, changes in profiles of proteins in different organelles, are those captured in such studies?
Okay, different expression of genes in organoids.
Yeah,
organelles.
In organelles.
Okay, that's a good question. That's kind of an area which is developing as well. Yes, we do some signals out of that, but how much of that signal is relevant only to that organelle is a very interesting question. There are papers coming out. We just have started working on those. It's a very good question, and there are methods which are becoming popular to get only material out from these organelles, which probably eventually going to populate in the whole field, and we are waiting for to understand those in a much broader way.
That's very fascinating, actually. How does expression profile from a heterogeneous sample look like, and how difficult is it to match therapy to it?
So heterogeneity means it's basically like what I showed in the, for example, pancreatic or colon cancer. So the patients with, for example, the stem-like subtypes have an entirely different gene expression profile, which before our study or similar studies, there were not information available. So that's how you identified the heterogeneity, but that's only coming from the gene expression heterogeneity. And these signatures associated with the drug response or anything would probably be embedded. For example, if that signature, there is a subtype called transit amplifying, which has indirectly EGFR signaling. That's why cetuximab seem to work there. So those kind of hidden information need to be really taken out by machine learning or other inferential statistics to understand them.
Okay. So there are a couple of questions where people want to know about the affordability of personalized treatments at this stage.
So affordability or applicable
affordability, like affordability.
It's a very good question. It varies from country to country. Obviously, for India perspective, probably I'm not the best person to answer, but there are certain therapies which are very, very expensive for Indian community to afford, and also maybe the tests associated with that. So from that perspective, it's very subjective and depending on the country. However, the whole point that we are trying to operate trying to help is also to see how we can bring down at least the cost of the test by doing different means of technology, bringing different means of technology, but long way to go.
So there's one question where the person wants to know that if the selected patients, they have lived under different environmental conditions and have different lifestyles, how much does this factor change the data that goes into the personalized medicine pipeline?
So it's a very good question, and also it's $1 million question. I mean, we are addressing a very subset, small stuff of it because it's a huge question. There are cases where environment, depending on even within India, I hear from others, and we are also trying to work is that depending on the regions people coming from, there are certain regions where people are more vulnerable to particular cancer versus others. However, globally, with what we mined, the breast cancer, for example, triple negative breast cancer at the gene expression level between the Caucasians and Indian community, for example, we do not see much of change in the biology there. But mutation rates and other things could probably be different, which is being mined by us and others.
Okay. So this question is, which is the most effective and easy method for detection of heterogeneity of cancers?
Sorry, there was some signal issue. Can you repeat that?
What is the most effective and easy method for detection of heterogeneity of cancers?
Okay. I wish I could answer that, but that's the difficult question. That's why we needed the machine learning. So that we have to, it's all trial and error. I wish I could tell one particular way to go and find it out. That's why these many tools and these many data and all those things are being generated.
Okay. So there's a person who says that he's working with H. Hello, can you hear me, Dr. Anguraj?
Yes, yes.
Okay. So the person here who is saying that he's working on H-TERT promoter region mutations, role in cancer progression. And he wants to know which software can he use for studying promoter mutations in case of cancer.
Okay. Actually, we work very, very little or probably nothing on transcription factors and promoters. So unfortunately, I'm not an expert in that.
Okay. I have a couple of questions asking that other than COVID, like are there a lot of studies, ML-based studies going into HIV research also? Would you have any information regarding that?
No, I mean, as I mentioned, I'm purely a cancer biologist. Again, in the COVID, I've worked only with the interest of patient response. With respect to virus, virology, I have no idea.
All right. So I think we will take a couple of more questions.
Okay.
There's like a stream of questions coming in, so I think everybody really enjoyed your presentation.
Okay. Thanks.
Yeah, this person wants to know that cancer requires a holistic treatment. So is it possible to compare integrative and complementary medicine so as to personalize the therapeutic regime for a person using machine learning approaches?
It's complementary medicine in a sense, like other
Ayurveda. I believe so.
Okay. I mean, again, I'm not an expert. I'm sure. I mean, there are experts in India who are really working on it, and I believe talking to them that there is a lot more opportunities, and we are trying to collaborate with them. I believe in that, but I have very little experience from that perspective.
Okay. So I have one last question. Is it possible that a person has some epigenetic changes in their genome, and when they undergo this change, so if epigenetic changes happen during the course of their treatment, and does the effectiveness of personalized medicine change during this period?
Sorry, can you repeat that again?
Yeah, so I think the question is that a person, is it possible that a person may go epigenetic changes during the course of the treatment in cancer, and does that affect the effectiveness of the personalized treatment so that they were grouped under?
I mean, yes, there could be possibility. There are data, some data available like MGMT type genes and methylation, all those things. But I believe there are not extensive information available that's in the clinic right now. But yes, epigenetic changes are also inevitable with the drug response, I guess.
Okay. Is it okay if we take a couple of more questions? Because,
sure, yeah.
Okay. Some students would like you to elaborate on the opportunities that you were mentioning with respect to COVID engine.
Okay, COVID engine is not our primary task, but because we started something on the side, and we are inviting community to contribute. So the opportunity would be that we have developed this COVID engine, which involves inferential statistics and machine learning. And there are, as you all know, there are loads of publicly available data that's pouring in for the COVID, and need of the hour is many things. How do we tackle this in multiple ways? Because it definitely affects the people and patients, and they're dying. But at the same time, it also affects the economy. So from the health point of view, there are a lot more we could contribute. But obviously, the one we are trying to do is from research and patients' response. So bottom line is, we have this COVID engine, we have some data, we have some ideas. So we are just trying to pitch into different sets of people who could contribute, and that's how we did this paper that we published. So any contribution is a good contribution. Obviously, if they come with an idea, okay, I read your paper, we have something that we could contribute, then we will explore through the consortium to see how that would be, that candidate would come in any form. We don't have a particular form. Whatever that would benefit for COVID. COVID is a good thing.
Yeah, that's really interesting to know, because especially even in today's time, when even the hospitals are having difficulty in managing, and like they don't, in India, for example, like, or anywhere, like how many beds are needed, and there's all these issues. So to be able to categorize with which one, categorize patients into different types, and the amount of care or the kind of care they need, I think it's the need of the hour right now. So
one of the major interests for us as a cancer biologist, just I wanted to mention, maybe useful, is that many of the collateral issues with the COVID, because of the COVID, many, not only cancer, other patients with pre-existing other diseases, they got deprioritized. So they're not getting therapies. So cancer is what our area, and I'm hearing this often, that people are deprioritized for therapies. So that means, how do we find who are really in a worse situation with cancer, how they need to be prioritized such that they would get the right treatment at the right time is quite important. And how do we find those patients is a major question. So we are also trying to work using the COVID engine. We have some idea, but obviously, that's not our major area. So we are welcoming people who are interested to contribute, so that that'd be useful for the community.
Thank you. Thank you, Dr. Anguraj. I think that was a wonderful presentation and a extensive discussion at the end. There was a huge flow of questions that kept coming. So, like I said, so I thank you, Dr. Anguraj, on behalf of the entire Manav team for taking out time for today's talk. So thanks a lot.
Thanks, Chavi, and thanks the Manav community and the participants, and thank you. Bye.
Thank you. I would also like to thank the outreach team at Manav for organizing the data science webinar series. The team will be shortly reaching out to all the participants regarding the feedback form and the participation certificates over email. Joining us next week is Dr. M.V. Krishna Shastri from National Center for Cell Science in Pune. So keep a watch on your emails and the social media handles of Manav to know more about our upcoming events. And we will see you all next week. Thank you.